Hi, so I'm, as mentioned, a computer scientist at Oceanit, and I'm going to be talking about some of our research currently ongoing with the DOE. We're currently about 4 months into a phase 2 project for detecting and characterizing roots in Minirhizotron imagery. First, introductions of the company. We're a small company in Honolulu that does science and technology research. We have about 100 people working at the company within a variety of disciplines. So I'm also working with some specialists on data processing, computer vision, and that for this project. In addition, we've got collaborators at the UC Davis, Dr. Patrick Brown, and Luke McCormack at the Morton Arboretum. And we'll be working later with CID Bioscience, who is one of the major makers of these devices to integrate into the software that they provide with it. So generally why? Minirhizotrons are these great little camera devices that can view into root growth by kind of having tubes in the ground and imaging the roots that grow around those tubes. And they provide a non-destructive view, which is great, so you're not destroying the exact stuff you're looking at, and can help kind of target nutrients and monitor health of the plants. So the problem here is that the analyzation of this imagery is time consuming. So you pull up the imagery, and then if you want to get statistics about the roots and where they are and how long they are, it takes a lot of time to trace all the roots. So automating this would remove this barrier, which kind of prevents them from being used more widely, while also allowing for faster, easier, more consistent scientific use. So here's an image of the roots. Our main problem, we've got these drawing all the lines and analyzing these takes a lot of time and can be imprecise because of that. And similarly, it can be kind of inconsistent if you have multiple people labeling. Everyone has a different view about what's too small to label and whether or not something is alive or dead or a root at all. So the goal is we want to create an automatic tool that will detect and characterize these roots so that we can have kind of some reference that can build specifically for each project to whatever they need and to share between researchers and say, we get the same results with the same eyes, essentially, albeit machine eyes. And then so the solution we're going for is to train a neural network to detect the roots and then use image processing to extract the root morphology, the traits of the roots, the branching factors, stuff like that. So the main challenge here is that automatically detecting roots is a hard problem. We have the issue of that the roots are sometimes lighter than the background, sometimes darker than the background, sometimes basically the same color as the soil. So you can't just use a simple color-based detection system like the thresholding here. There is no threshold which you detect all the roots and don't detect a lot of these various either background or artifacts within the tube, moisture, scratches, stuff like that. And so the need there is you want something that can actually analyze the structure. And that's where we come into neural networks. So just to give a general background, the broad process for training is that you want to collect a bunch of data. You do some data preparation to throw out bad images, to extract features or otherwise enhance the images to show what you want. And then you split this data up into different data sets. So you have training data set, which is the data that most people think of, goes into the net, churns a lot, generates a result. Validation data is kind of like an in-process testing. And then you have a final set called testing data that you really want to keep aside so that you know that you're actually learning, because there is some potential that if you don't have kind of some separate eyes, your validation of the results can be biased. You can overtrain, you can get something that just memorizes your data set, doesn't learn the problem. So then we push all that into the model, turn a bunch on GPUs, generate a lot of heat, and get some results. Then we can compare that against the testing to verify that it is actually learning the problem, not just the data set. And then we potentially go back, refine our hyperparameters, which is all the traits you decide in the model, the size of it, learning factors, stuff like that. And also go back and look at our data collection, data preparation. So maybe we don't have enough data, maybe we didn't highlight something, there's some better way to differentiate things. So in particular for image data, what we're looking at, convolutional neural networks are kind of the gold standard. And they're designed for structured data that particularly has some sort of spatial relationship. So images, the pixel next to one thing is related to the pixel one step over. They use these features to first learn at the very low level pixel level, stuff like lines, where colors are, stuff like that. And then you can step upwards and then apply it again on those features to get higher level features. Build shapes, from shapes you build larger structures, and then finally you can get some end result. Typically we apply these to image patches so that the result you get is independent of exactly where it is on the actual picture. If you have a root, for instance, in the upper left corner, it's no different from a root right in the middle of the image or in the lower right corner. So the kind of looking at this problem, we have this issue of these are very difficult to see things that are blended in the background, very low contrast. And that immediately reminded me of a use of a technology called UNETs on eyes and retinal images. So we have the veins, they get dimmer and dimmer as they get smaller, but we can still pick them out through this technology. So this is a type of convolutional net that basically steps down, gets more hierarchically complex as you go, but then when you're building it back up, you produce a mask image. So you've got an input image and a mask image, and you train it to translate between the two of them. And it has these kind of orange lines here that are called skip connections that allow it to learn a little easier in this sort of type of generation. And so they've been used in biological images and in other studies for trying to detect roots. The main problem we have is that for the images, we have, in say the eye example, we have an image and we have a mask, and that's our goal and what we want to predict. But for roots, we don't have a mask. We have an image, and then most of the existing data is in various software packages. It's just lines. So lines tracing roots, because that's what people are primarily looking for when using these, is stuff like length. And you can include width in that, but it's not always common. But it's not a pixel by pixel drawing of where the root is. So we have this issue of we need to somehow figure out how to take this existing data and somehow get a mask to generate for our training process. So one of the solutions is to ignore the existing data, just draw new masks for everything. And there's been some research that has used this to get results. And the main problem with this is that this is a pretty complex image. Really drawing pixel by pixel where all of these roots is even more laborious than doing the line tracing. And you run into the issue of you can maybe make a data set that will work for your particular data. You take all your data and you trace 50 to 200 images, depending on which solution people have tried. And you can apply it, but there's limited guarantee that you're actually learning the problem, because the smaller data set you have, the more likely there are to run into overfitting. And then furthermore, it's not transferable from this data set to another. So these are also all root images with wildly varying backgrounds, wildly varying colors. And so you can't just take a model that you made. So you put a lot of effort in, you trace 200 images. You can't just pick it and add it to, bring it to something else. And from talking to people who have tried this, you actually can't even necessarily bring it from just one field to the next, because it's likely running into that overfitting problem where it's learning one environment too closely and doesn't generalize into other environments. So what we want to do is find a way to tap into all this great existing data that we have and still be able to generate the models to make predictions. So the kind of solution to this is that it's built into neural networks already that the data isn't perfect. We already expect that there is labeling errors, that people make mistakes. Even when you're doing something that's just 5 choices, sometimes people click the wrong thing. But the whole concept is that through a lot of data, we kind of wipe away those mistakes. We learn the general patterns that exist in that data, and then can produce results from it. So even though we have this data, on the right side that looks like it's not really on the root for a part of it, we can kind of make a mask from it anyway. So instead of having a fixed, this is a root here, definitely we have a probabilistic mask where the farther you get from it, the less likely it is. And so we get this kind of halo around the root that covers most of the root. And you can see that usually the root is masked as root, and that most of the soil is unmasked, which is great because then in the end, we're kind of pushing it to say like, okay, I need you to detect soil and root. Those are essentially the two things we're looking for. And most of the soil is unlabeled, otherwise marked as soil, despite that there being some that is marked under the root. And then similarly, most of the root is marked as root, even though some of it is outside of that pattern. And so throughout the problem that we're presenting it, we're kind of saying, okay, look at this little small patch and say, is this root or soil? And when you're looking at a root, you're going to be able to tell some feature that's going to be different from the background. When you're looking at soil, it's pretty much the same as all the soil elsewhere. It's not really a different type of thing. And so the broader soil is going to be able to say, no, that's soil. So that said, we would prefer we didn't have a whole lot of imperfect data. The more you get, the harder it is, the more data you need, and the more risky it is that you're going to get overfitting and need to do some changes to the process. So we have a few different errors that we want to look into and we can correct and make our process better. One here is the example of people will copy roots from one session to the next because the whole point is that the roots grow. And most of the time, a root that was there in the previous session will still be there in the next. But the devices that put down, sometimes the way you put them down changes exactly where the positioning is, and you get these kind of root shifts. So if you copy them over and then don't do the fixing to say like, oh, actually the image shift, we need to change everything, you get these markings that are off. Similarly, you can get some errors where you put the device down and into a tube, but it actually you end up duplicating one of the levels or skipping one of the levels, and then everything's off by one. So this thing really should be at the bottom and to match all the rest. But if you look at it without doing any fixes, you just see the data go kind of rapidly change from one collection to the next. The nice thing is that we can solve both of these with image alignment. So we take all the images and we find the traits in the soil and the root to create transforms that will align all these images so that they stack up, and basically the roots stay in the same place, meaning that if you copied roots, it will stay in the same place. We can note when the roots markings haven't changed, but the background has, and we can fix them. And then similarly, the alignment process will find these misaligned kind of image ordering and allow you to correct so that you either delete a duplicate or you kind of add a buffer skip so that everything matches back up. So once we have that, we have the data, we need to kind of divide it for our process. So we don't want to take full images because it's not important that we have full 4,000 pixel images. We want to take kind of small little patches so we can learn the kind of smaller low level root dynamics. So we split up in all these overlapping boxes so that nothing is kind of on the border. We don't chop any root up, so everything will have some chance to be seen. Then we discard all the images that are just soil because we have more than enough soil in all the other images. Most of every patch is soil. And then with the roots that make it through, we split it into training and testing sets. And we split it by, for the studies that we have where we have multiple tubes, we split it by tubes so that we're sure that we don't have learned specific root structure. So we want to recognize roots in general, not that there is a root that looks, has a particular bend in it. And so we split them up so they can get kind of a clean testing that would indicate whether or not we can be successful later on when we apply it to data that we haven't seen it at all. So once we have all these little patches, we push it through our big machine. We run the training for basically a number of epochs, which is you run through a little data set, update your model. Essentially, this is kind of like a big, like a line fit or polynomial fit that the bigger, the more generally you make it, you could fit things perfectly, but you want to kind of tune it to be the right size for the problem you're solving. And so we push it through our system. We have some metrics and methods to try to prevent it from overfitting. And then we get the predictions. And so you can use the predictions and then find out how you need to correct the system to get closer to where the result should be. So in general, each run of training of this is like 48 hours for a very large data set. And then evaluating the network afterwards is very quick. To like 10 seconds per image. So you could get your results on an image before you walk to the next place, next tube to get to gather a new one. And so here's some examples of our results. On the left column, you've got the raw patch. We've got the user's label with our kind of little fuzzy halo around it, an overlay to kind of show where that is, and then our prediction and the same overlay for our prediction. And you can see in these that not only have we kind of fixed the position of some of the labeling that was off, that had drifted and wasn't really on the route anymore, but we've also detected some routes that weren't marked at all. They were just missed. So that's kind of a good indication that we are learning the problem in general rather than learning our data set, and we can find new things and improve even on what human labelers had been doing. So we had these great results, and then we wanted to test to make sure this actually worked in others. So we applied the exact same methodology to two other data sets, one with the same species, but a different kind of soil and camera system, and another with just a variety of other species. And both had similarly good results for locating where the roots are, finding new roots, that sort of aspect. So this is great. It's kind of a good indication that this can expand further, but we always want to make sure we can make more general sets and make some claims that this will work on everything. So with this, you've got, once you have your alignment and then you have your root markings, you can watch the roots grow over time. It's new roots appear, old roots kind of can disappear, and we still maintain our tracking. And so we can have a view of this root grew, not just one image and the next image and the next image, and you have to make your own decisions. So with these, from the neural net, since we trained them in these kind of like halos, they had kind of a fuzzy pattern of like, this is where the root is. And then we want to take those and actually reduce it down. It's something that can give us measurements, particularly the length of the root and the width of the root. To do that, we kind of take the fuzziness and then we squeeze it down into its center area. And from there, we can fit a Bezier curve to get kind of a really fine controlled fit on the root. And this is even better than the line segments that are frequently elsewhere because, well, most roots aren't perfect straight lines. So this allows it to have a good curve and then get a nice measure of the fit along the way. Once we have these center lines, we then can look at to find where the width of the root is. So we have a pretty good kind of guess of that from the neural network, but this is where we can kind of inject human understanding of the problem. And from the center line, we can step out bit by bit, kind of pixel by pixel, making a wider and wider path, and look for when eventually we get a big change in the color under that's covered in that area. And so eventually you get to the edge of the root, even though there's not necessarily a defined, all roots are lighter, all roots are darker. You can find some point where one more pixel and suddenly things are starting to change. And there's always some aspect of kind of like a slope, and you can kind of fit in there and know that this is where the root border is. So with that, we do all these results, we get these things. We want to check, well, okay, how do we actually compare to the humans? We don't want to say, okay, we looked at some pictures, everything's perfect, good, sell, use it for all your important research. So we compared it to the root fly, which is the labeling software length. And for basically, we looked at what the length of all our roots was and the length of their roots were. Because even though there's kind of inaccuracies in where the drawing is and things shift, the goal is to measure roots. And so that was kind of the strongest measure of from these labels. And so the great news you can see along this is that everything's clustered along that y = x line, which means you've got pretty good agreement. We've got a few ones that seem to be way off in the stratosphere and either indicated that we had a very poor measure of the thing, or that there may have been something wrong with the data, which it turns out, yeah, there's frequently various errors from people doing this as they sit for hours on end and draw lines on screens. So here's one of the outliers. Something went wrong. I don't know what was happening. But this one particular frame had what looks like a maybe a frustration drawing or maybe just a mistake. And we have all these back and forth overlays of the same lines. So the one before and the one after, perfectly fine. So we can accept that some of these are going to be a little messy, and then, but the broader data set will allow us to carry through. So for instance, in comparing like the data that we saw for various things, we have some data sets that are thousands of images. Our smallest data set is about 500 images. So we're able to kind of see it in a range there. And the more you have, the more you can have a few that are really just terrible. So this is all working towards what our phase 2 work is. We're about 4 months into the project. We have a 2-year research window that we're working on. And the goal is to not just do this and say, great, it works. We want to actually make something that people can use so that people can take their data sets and automatically mark them and do the characterization. So generally, our goal is that we want to use the stuff you use, so that we don't want to say, okay, well, we're going to make a new tool. You have to learn a new tool. If you're using one of these existing tools, great. We can ingest the images that you've gathered from them, and we can make either train a custom model for you, because we believe that if you have a custom model, it's probably going to be your best perfect fit. Or if you want to instead kind of have a comparison model or use a general model, you can use a model that someone else has developed, either us or another researcher. And then once we have this and we've run the model against it, done detections, we provide all the information. So we can show you the root length, the width, the branching factor, when roots die, and provide all that back to you so that you can then do whatever analysis you need to do. So the benefits here is that, as I was saying, it takes about 10 seconds to do labeling, what used to take minutes to hours, depending on the complexity. It can be more consistent than humans because we've got a potentially single model that can do the analysis. You don't have to worry about if the person that did labeling last year is still available to do labeling for the next year. Or if you want to make comparisons of different studies, you have a single point that you can say, okay, we both ran it through this and had similar results, or we had different results in this way. And so you can trust that it's not just a labeling difference, it's some actual difference. And then finally, it's often more accurate than humans because this is a long, tedious task. There's errors in collection that are not unnoticed, and people will make mistakes. And so we found, at least in our existing examples, that we're seeing cases where we mark roots that the user did not because the labeler just missed them. I mean, there's a lot of roots to mark in this, and that's the challenge of the problem. So the cool thing though is that it's not necessarily just for roots. So we have some interest within our collaborators. They want to know kind of where are rhizomorphs, so other root-like structures. And the process in general is not only devoted to roots. You can do it to other sorts of imagery. So if you have imagery and labeling of rhizomorphs or root nodules or other structures, then you can do the same segmentation process, depending on if you're breaking from the root, like a root nodule. You might need to have different processing modules afterwards, kind of find spheres rather than lines, but that's a simple addition. And then you can also see stuff like kind of root hairs or dispersed fungal areas that aren't really a defined thing, but it's like this area has issues. And then similarly, the kind of main thing on all of this is what sort of data is available. So tracing or patterning or kind of some sort of box is the best, but depending on how far you're straying from kind of existing structures and whether or not we can kind of key in on things like color for, say, like the color of a root nodule compared to the color of the root it's attached to, we can kind of work and try to translate other things into something that will work here. So as I'm coming to the end here, I have one general plea. If someone is here watching this because they have used Minirhizotrons in the past and have labeled data, please help us make this better. We want to make a system that we can trust to be work in a general case, so that if you have a new crop or new plant, it's not going to just fall apart. And we ideally want to make something that's a general model so that not everyone has to go through a labeling process to be able to use these devices and get results to use in science or to monitor plant health or whatever they want to use it for. So if you have anything, please contact us. We'd love to see it. And with that, it's the end of my talk. And I'd just love to entertain any comments or questions.
Great, thank you very much, Zach. That was an excellent talk, very informative. So I just start with one question. So correct me if I'm wrong, but my understanding was that most of the data you mentioned was derived from Rhizotron studies for root phenotyping. So is the technology, the machine learning technology that you described, can be employed for any other data derived from other technologies, like X-ray tomography or other similar systems for root phenotyping in soil?
Yeah, absolutely. So the project that we're focusing on is from Minirhizotron data and detecting things in those images. But machine learning in general can handle almost any problem as long as you have some data and someone with expertise to make sure that you don't make egregious mistakes with that data. And so the kind of the farther you get from the imagery from Minirhizotrons, you might kind of need to do different things on the image processing aspect. But the general process of the mask to, or the image to the mask, as long as you have something that you can kind of estimate a mask or produce a mask like we do, you can do that sort of learning with any sort of imagery or even higher structured data. We've worked with hyperspectral data before or other sort of 3D renderings of things.
Great, thank you. So the other question is that, so your software only works on 2D images, not on 3D images?
So, well, yes, because that's the only thing we can get out of our kind of target sensor. If there was, we could do similar things with 3D images, but for the purpose of this research, it's to try to support the Minirhizotron type imagery.
Great. So another question would be, so I just got another here. So it's actually, yeah, the question is that root morphology, soil mapping to ML modeling prediction explained well with rhizomes. Can this help in improving some medicinal plant health, prevent infection?
We hope so. So yeah, that's one of the benefits that we see in being able to have something of this nature that is less laborious so that it can actually be used in a broader audience, is that you can use them for a wider field essentially. So that in the same way that we can detect the root length and width and all that, we can also detect when roots start to die or when roots aren't growing as much in one way or another. Or say if we have kind of an additional detection for like funguses or other things that we believe to be an infection. So you can see kind of in your data sets where the plant roots are starting to die off, and that can sometimes be before you'll see that effect elsewhere. And so you could have kind of, depending on how many of these you wanted to do for the data collection, because the data collection also you have to spend some time to take the images, though much less than the labeling effort. You could either put them as samples in various plants in some kind of shared plot, or you can have the one per every plant if it's usually for something that's like a nut tree or something. There's more value in having individual views because it's a longer term growing prospect.
Awesome. Okay, next question. Does the camera only operate in the visible spectrum? Are there hyperspectral images available that may hold additional information?
Yeah, that's a great question. So the ones that are in use now are only visible spectrum. They're actually kind of modified from like flatbed scanner technology and then kind of put into a tube and twirled rather than scanned across. But the both CID and some other researchers have been interested in changing it into a hyperspectral or multispectral camera. And that hopefully should be able to make this process a lot easier and have a lot more information that can be learned from those pictures. And it's a nice thing because the way these cameras function is they're down in a tube and a dark tube. And so all the light that is being displayed and picked up by the camera are the light that you provide. So it kind of has a natural aid in being able to say, okay, we want to take a photograph at this wavelength spectrum. And then we take another one at this wavelength spectrum, and you don't have stray light coming in and making it harder for you. So I hope to see that. I know I've seen some kind of like custom made devices, but not in the broader, you can just go to the website and buy one sort of a situation yet, but hopefully soon.
Wonderful. So next question. So great talk. How hard is it to discriminate between plant species in the same Rhizotron image? And another part of the question, or use of optodes on the Rhizotron tube.
Sorry, what's the second part?
It's the use of optodes on the Rhizotron tube.
Okay.
Or maybe you can focus on the first part first, or how hard is it to discriminate between plant species in the same Rhizotron image?
Yeah, so it's hard. The main difficulty is in having data that says this root is this species, this root is another species, because that's not as common of a research question. So there aren't as many existing databases with that. So one of the databases we tested is actually one of the challenges it introduced was that Martin Arboretum only wanted to mark the data from the target species that we're looking at. So at the kind of upper levels, there was a lot of various other plants that had roots in the images. And so we found that with this is ongoing, so we're trying to tune it and solve this ideally without having to do another labeling effort. But we found that the target species was we were able to learn that very well because that was all marked. We had things fine to say this is the type of thing that you want to mark and this is what you want to find. The non-target species, the other root type species, had much more of a kind of a mixed bag. So it wasn't that it was just like we find roots and we all, any type of root is found because we can't distinguish between the target and non-target. But there were kind of probably a toss up 50-50 sort of situation of some non-targets would be classified as roots and some non-targets would be classified as not necessarily soil in this, but not of interest. So that is one of the problems we want to study and try to tackle because we would like to be able to provide that idea, especially with, you've got another plant growing here. That's, that might not be what you're hoping for. But the main challenge there is finding data sets where someone has gone through the effort to not only mark all the roots, but mark them and note that this one is different from this one. And we have a little data that has that sort of a thing, but not as plentiful as what we've used for kind of the broader detection problem. And then, so the second question, I can't answer that. I'm a computer scientist. I'm not as familiar with the terminology.
Okay, so yeah, that was kind of more explanation of that, but yeah, we can definitely have discussion about this later. But that was mainly on following up multispectral imaging question. There are oxygen optodes that are used to measure oxygen availability in soil. Can these be coated on the Rhizotron tubes?
Oh, yeah, I think that would work pretty well. I can't say for sure if the coating might disrupt some of the imaging, but I imagine that you should be able to do it in some way to kind of bring that out. And yeah, similarly, if you have other things that you want to find in the soil and can do something either with just visually, if it's obvious to see, or if there's some aspect of either hyperspectral imagery or some sort of thing to kind of bring out a reaction, then you could just as easily perform this process to recognize, mark, and kind of characterize all of that as well.